Hello, everyone. A very warm welcome to you all, and thank you for joining us for today's presentation. I'm John Norman. I am Head of Marketing here at Optibrium and moderator for today's webinar. And well, it's great to see so many people joining our program here today, which, as you can see, is titled A Global Deep Learning Model for Global Health Drug Discovery. And we'll be describing the generation and validation of a global model using deep learning imputation on a data set of 300,000 compounds and 500 experimental endpoints targeting global health indications. So now it's time to hand over to our presenters, who today are Samar Mahmood, Senior Scientist at Optibrium, and Matt Siegel, who is our CEO. Matt, I will now hand things over to you.
Thank you very much, John, and thank you all for joining us today. It's great to see so many of you here in the audience. It's my great pleasure to introduce my colleague, Samar Mahmood. Samar has a background in drug discovery and pharmacy, and she completed her PhD in cheminformatics and toxicology in Andreas Bender's group at the University of Cambridge. Immediately after that, she joined us here at Optibrium in 2018, and has led several projects applying our unique technologies to drug discovery projects with our collaborators and customers. And today, Samar is going to share the results from her work on this particular project, which was undertaken in collaboration with the University of Dundee. Samar, before we get started though, you've worked on a wide range of other projects using the Alchemite method that we're going to be talking about today. I wonder, just before we get into the main body of the presentation, if you could say just a few words about your experience using Alchemite and some of the other projects you've worked on.
Yeah, thanks, Matt, for the introduction. Yeah, so I've been interested in applying computation methods to facilitate drug discovery, and in particular in combining machine learning methods and expert knowledge, not only to build predictive models, but also to dig into the data and mine the data in order to gain some knowledge and insights. So since I've joined Optibrium, I had the chance to use Alchemite and apply to a range of different projects, including predicting kinase activity, sensory properties, and in vivo efficacy tests. And as you may know, that many of the deep learning methods are actually act like black boxes. So it has been nice to see how Alchemite can give not only high accuracy, but it also helps us to get insights into the patterns in the data. So yeah, so overall, it has been a pleasure working with our collaborators and understanding their projects and the challenges.
Thank you very much, Samar. And actually, for some of those projects you just mentioned, I'll just let the audience know that there's more webinars coming in the near future, so keep an eye out for those and you'll gain some more insights. But today, we're going to be focusing on a collaboration with the University of Dundee. I'm just going to kick off with a quick introduction to deep learning imputation and the Alchemite method in particular. I'll then hand over to Samar, who's going to tell us about the data set that she was working with, the objectives of this particular project, the validation of the resulting models, and then give an illustrative example of how the resulting model was applied to a particular challenge in the optimization of an anti-TB therapeutic. And we'll come back at the end with some conclusions before going on to the Q&A session. So as I mentioned, I'm just going to give a high-level introduction to this idea of deep learning imputation and the Alchemite method. I know that many of you in the audience have been to our previous webinars, so this will be quite a high-level introduction, but you'll see the references at the bottom of the slides that have all of the details regarding the underlying method, its validation, and benchmarking against other methods. So just by way of introduction, I wanted to start by defining this idea of imputation and comparing that with more conventional predictive models. We're all very familiar with predictive models, for example, QSAR models that take a set of input features. In our particular case, what we do is we calculate descriptors for the compounds that we're interested in. So here we've got the sort of matrix of compounds down the y-axis and descriptors across the top here, and we can calculate every descriptor for every single compound to give us a complete matrix. We can feed those descriptors, those features, into whatever modeling method we want to use, whether it be random forests or SVMs or Gaussian processes or deep neural networks, whatever that might be. And then we can generate predictions for one or more experimental endpoints, shown here by these purple squares on the right-hand side. Now, imputation is quite different. Imputation actually starts with the limited experimental data that we have available, shown here as sort of assays across the top. And one of the things we know about drug discovery is that this data is very incomplete. It's very rare that a compound has been measured in every assay that might be of interest, and it's very rare that every compound has been run in any particular assay. So illustrated here by this sparse matrix where the green points represent measurements that have been made and the white space represents that missing data that we haven't yet measured. And what imputation does is it uses this limited data that are available to fill in the gaps, the purple squares again here on the right-hand side. Now, the particular method that we're using and going to be talking about today is called Alchemite, and Alchemite is a collaboration with our partners at Intelligens. Intelligens originally developed this method in the material science domain, got some fantastic results in a range of different applications there, but we work with them exclusively to apply this method in drug discovery and small molecule optimization. Now, Alchemite can take the usual sorts of descriptors as input, so we can still take these calculated values, and that enables us to capture that structure-activity relationship between the compound structures as represented by the descriptors and a variety of different experimental endpoints. But what it can also do is take this sparse data directly as input. What that means is, even though most of that data is missing, it can actually learn directly about the relationships between these different experimental endpoints, those correlations that we know exist in our data, as well as the structure-activity relationship. And this makes much better use of that sparse and noisy experimental data than conventional QSAR models. So it can fill in the gaps, the purple squares here on the right-hand side, filling in the gaps between the actual measured values. And something that Samar will also show you later on in the presentation is it can also make predictions for virtual compounds. So where we have not yet made or tested a compound, of course we have no experimental data, we can still just use the descriptors as we would in any QSAR model and make very high quality predictions of the likely properties of those compounds. And these enable us to target high quality compounds for our projects. Again, if you want to know more about the methods, I've referenced a couple of our publications down here in Journal of Chemical Information and Modeling that provides much more detail and examples and benchmarks and so on. The other feature of Alchemite that we're going to focus on today is its ability to estimate the uncertainty in each individual prediction. So Alchemite doesn't just produce a single value and say, here's the predicted value for this measurement, for this compound. It actually generates a probability distribution for that value, sort of illustrated here by these purple sort of histograms. What that means is that sometimes we can see these sort of distributions that are very broad and smeared out. And this is the model saying, the value's somewhere around here, but I'm not really sure. I haven't got enough information. And so it's a very big error bar on that predicted value. Whereas in other cases, we can see very sharply peaked probability distributions. That's the model saying that it's confident in that value. It's got a small error bar. And of course, it's the examples like the ones on the left-hand side that we're most interested in, because those confident values enable us to most accurately target high-quality compounds for our particular project objectives. On the other hand, where we see a lot of uncertainty, like the example on the right-hand side, that's where experimental data will add the most value. Where we've got the greatest uncertainty, measuring some of those points will actually give us more information to make better predictions, and so it helps us to prioritize our experimental resources as well. So with that little bit of background, I'm going to hand over to you, Samar, and Samar is going to take us through the data set and the objectives of the projects, followed by the validation of the resulting models. Over to you.
Thanks, Matt. Yep, so we'll be describing the results of applying Alkamite in a project in collaboration with the Drug Discovery Unit at the University of Dundee. And the Drug Discovery Unit has high expertise in running high-throughput screening, medicinal chemistry, and computational chemistry, but their main focus is to develop drugs for neglected diseases. So the key objective or key goal of this project is to predict TB activities and ADME properties with high accuracy in order to guide optimization of compounds in a specific project. And in order to do so, we've compared multiple models. So we've compared the performance of models that build on the project-specific dataset versus models that were built from data collected from multiple diverse projects. We've also compared the performance of the imputation and virtual models on this dataset. So the dataset that we've used for this analysis includes the global dataset, which includes 300,000 compounds that has measurements against 468 experimental endpoints that covers several developing world neglected diseases areas. And it's worth mentioning that only 3% of this dataset is complete, which means 97% is missing. And a subset of the global dataset is the project dataset, which corresponds to a single TB project. And this dataset represents around 500, just under 500 compounds measured against 34 experimental endpoints, and 40% of this subset is complete. So just to give a quick illustration of how the data looks like. So here we represent the global data set where we have compounds in rows and the assay endpoints in columns. And a subset, just a small subset of this data set is the project data set, where a small fraction of the compounds are are represented as the project-specific compounds. And it's also worth mentioning that these compounds are only screened against the endpoints within the project data set. So in this analysis, we built two types of models, the imputation and virtual Alkamite models. So the imputation model, as Matt has explained, it generates predictions for test data points using sparse assay data as input, as well as molecular descriptors. And we've built these models to assess the Alkamite's ability to fill in the gaps and generate predictions for the missing gaps in the matrix of experimental data for compounds that have been synthesized and tested for some of the assays. Whereas the virtual model, these are built to expect only molecular descriptors as input at test time. And we've built these models to assess the Alkamite's ability to make predictions based only on compound structure, which means we're assessing how Alkamite will predict compounds, activities for compounds that have not been synthesized or tested. So the process of building the imputation model is we start with a matrix of sparse experimental data, as represented here, where we have the compounds in rows and assays in columns, and the green squares represent the available measured values. So we split the data into training and test by compounds. In this case, we've applied an 80-20 split. We use the training set to build the model, where it would use the descriptor matrix to understand the structure-activity relationship and also explore the inter-assay correlation within the assay space. Then the model is applied on the test set to make predictions and to impute values for each of the measured values. And in order to do so, if we take the red square here as an example, the model would use the descriptor vector as input, as well as any experimental data points available for that compound. Then after we generate the imputed values in the test set, we go back and compare any available experimental measurements in order to assess the performance of the imputation model. Similarly, for the virtual model, it is built on using the training set, using both the descriptor matrix, as the brown squares here, as well as the assay, where it would learn both the structure-activity relationship and the inter-assay correlation. However, in this case, we tell the model not to expect experimental data at test time. So when the virtual model is applied to the test set, it would use only the molecular descriptors as input. And then the generated predictions in the test set are compared with any available experimental data for the compounds in the test set. And that enables us to assess the performance of the virtual model. And just before exploring the results, I'm going to briefly describe how we do the assessment of the results. So we assess the performance of the Alchemite model in two main ways. One way is a high level, looking at the performance overall, and we use in this case the coefficient of determination, where we measure this metric for each endpoint. So the R-squared or coefficient of determination range between -infinity to 1, where 1 represents 100% accuracy, and we can use these calculated coefficients of determination to generate a performance profile, such as the plot here, where we rank endpoints based on accuracy from left to right, where the most accurate endpoints are towards the left. And by looking at the profile, we can understand how well the model performed on this data set overall. Generally speaking, the larger the area under the curve in this profile, the higher the accuracy. And a way to summarize the profile is looking at the median R-squared across all endpoints, and this gives us an indication of how the model performed on the data set. A second way of analyzing or investigating the model performance is looking into the performance on individual endpoints. And in this case, we use the RMSE, the root mean square error, as a metric. And because the Alchemite model generates uncertainty estimates for each prediction, we can rank the predictions by confidence, as we see here in the x-axis of the plot, where the most confident predictions are towards the left, against the observed error in the y-axis. So for 100% of the predictions, we observe the highest error. And as we move toward the left and we focus on the most confident predictions, we see that the accuracy increases and the error decreases. And what this tells us is that there is a strong correlation between the uncertainty estimated by the model and the observed accuracy. So next we are going to look at the results from model validation, and we will be starting with results from the global model. So the plot here represents the performance profile of the global imputation and the global virtual models, where the imputation in the orange line and the global virtual model is in blue. And what we see here is that the global imputation model significantly outperforms the global virtual model, where we see that the median R-squared is higher for the imputation model, and also at a given level of accuracy. So for example, if we set an accuracy of an R-squared of 0.5, we see that the number of endpoints that were predicted with that level of accuracy is much higher using the imputation model in comparison to the Alchemite virtual model. Actually, in this case, it's 4-fold increase. And this is quite interesting because the global data set is quite sparse, so only 3% of the experimental data is present, which means that that the Alchemite model would use and leverage the inter-assay correlations in order to improve the predictions of the model, even if these data set is if these experimental data are sparse. Secondly, we can look at the performance of the model on the project test set, and in this case, we would compare 4 models, 2 global models and 2 project-specific models, and for each, an imputation and virtual models are presented. So the profile here shows the performance of the imputation models represented in orange for the project-specific and blue for the global model, and the black for the global virtual and green for the project-specific model. And again, the same pattern as we see before, the imputation model significantly outperformed the virtual model in both for both the global and project-specific models. But what is particularly interesting is that the global and project-specific imputation models are quite similar in their performance, and they're achieving similar median R-squared. And when we compare the global virtual model, we see that it outperforms the project-specific virtual models, and that's because it explores a wider range of chemical space and learns the structure activity relationship from a larger number of compounds. So next, we can explore the performance of the Alchemite imputation model on individual endpoints. And we've selected some examples of endpoints of high interest. And in this case, we've selected one of the TB activity endpoint, where the model performed very well actually on the test set of this endpoint. So it achieved an R-squared of 0.81. And that's also demonstrated by the scatter plot of the observed value versus predicted values, where we see that the points roughly are very close to the identity line. And for 100% of the data, we see that the error bar is roughly around 0.3. But as we focus on the most confident 50% of predictions, we see that the error bars become smaller, the data points become closer to the identity line, and that is also reflected to a smaller value of the corresponding RMSE. We can even focus on the most confident 10% of predictions, and we see that the points become really close to the identity line, and that is also reflected in a smaller RMSE. And this shows the strong correlation between the model confidence and the observed accuracy. It's quite interesting also to see a couple of data points where it's presented as outlier, where the model was really confident about these predictions, which means that we can further explore these compounds experimentally to make sure that the measured values are accurate. And that actually these outliers are responsible for the uptick in the plot that we see here towards the left. We are going to look also at another example of an endpoint, the hepatocyte clearance, where the model has not performed very well on the test set. So the R-squared in this case was around 0.31, and that's also reflected here in the scatter plot, where we see that the data points are more scattered around the identity line, and also we see a large error bars around many of these points. So for 100% of the data, as shown here in the scatter plot, the corresponding RMSE is around 8. But when we focus on the most confident 50% of predictions, we see how the points become very close to the identity line, and we see also a significant drop in the RMSE value. We can also explore the most confident 10% of predictions, where we see the points are really close to the identity line, and the corresponding RMSE also drops to 2, which shows that the strong correlation between the estimated uncertainty and the observed accuracy. So even when we have a model that has a poor overall performance on an endpoint, we don't have to throw this model overall, so we can still identify the accurate predictions and use that with confidence. So to summarize the results so far, we've seen that the global and project-specific imputation models had equivalent performance on the project-specific data. And that means that we are not losing accuracy from including unrelated experimental endpoints and chemistry as in the global model. We've also seen that the global virtual model outperforms the project-specific model because the global learner model can learn from additional chemical diversity. So what we actually learned from this is that we can build a single model across diverse data set covering multiple therapeutic areas and experimental endpoints. So we would save time because there is no need to build multiple individual models. And we could also maximize the gained information, where we can learn the inter-assay correlations across multiple project chemistry and therapeutic areas simultaneously. We've also learned that the uncertainty estimates generated by Alchemide can be used reliably to identify the most accurate predictions. And we can apply these uncertainty estimates to make predictions based on the most confident results. And I'm going to hand in a moment to Matt, where he will explain how we are using these uncertainty estimates for decision making in this particular project. And also we can apply these uncertainty estimates to avoid missed opportunities by rejecting compounds based on inaccurate experimental data. So I'm handing to you, Matt, so that you can describe to us how multi-parameter optimization can be used in the context of this project.
Thank you very much, Samar. And so we've seen how the models were built and the great validation results. So I'm just going to introduce a little bit about how we can use those models and these estimates of uncertainty in the context of a drug discovery project, in this case, a particular TB project being undertaken by the University of Dundee Drug Discovery Unit. So like all drug discovery projects, the project was not interested just in active compounds, but a balance of activity with good physicochemical and ADME properties. So here's an example of the profile. So you can see they were looking for compounds that were active actually in 2 different assays for TB activity, not just 1, but 2 different assays, as well as good solubility, both at pH 7.4 and in FASIF, avoid high mouse plasma protein binding. Mouse is the first animal model that's used, and also good metabolic stability, both in mouse hepatocytes and microsomes. And I guess in common with many projects, they were finding a challenge achieving a balance of activity with both hepatocyte stability and solubility. And the strategy here was to explore a large virtual library enumerated around the particular series of interest to this project. Wanted to test whether these balance of properties were accessible within reasonably synthesizable compounds around that particular series. And so what Samar will describe is how this global virtual model built using Alchemide was applied to this large virtual library and test whether there's a high probability or any significant probability of achieving that balance of properties. Now, those of you who know us at Optibrium will know that we're strong fans of the idea of multi-parameter optimization. And so just to introduce how we'll apply these models, we're going to assess the predicted values against the profile that I just showed you. So our probabilistic scoring approach enables you to define a profile of properties for a particular project. You can say, what are the properties we're interested in? What are the criteria that we're trying to achieve? And also, what is the relative importance of each of those individual outcomes? We know that some criteria are critical, for example, activity, while others we might be willing to compromise or trade off. So you can define that balance and that trade-off you're willing to make. It brings together all of the data. In this particular case, we're talking about predictions, but this can also be applied to experimental data as well. And it assesses the compound's chance of success against this profile, taking into account not only the values that are predicted or measured, but also that uncertainty. So you can rank them by their likelihood of success and also drill down onto the individual compounds and see the impact of each property on that overall score. In this particular case, you can see the couple of low bars here are the biggest potential issues, and they correspond in the key to log P and hERG inhibition. So it provides that guidelines as to what are the critical issues that we need to address. Now, just to go a little bit further, I showed you this example of a profile where we have our criteria for each property, but actually underlying those criteria, it's not just a hard cutoff of sort of good or bad, but actually we can represent more subtle trade-offs by what we call a scoring function, or often known as a desirability function, that links a compound's likelihood of success to a particular value of a property. And so what we can do is take our property data, again, it could be experimental or predicted. In this case, we're looking at predictions, assess it against our criteria for success, these criteria and desirability functions and their relative importance, taking into account all of that underlying experimental or statistical uncertainty to calculate the score, which, as I mentioned, is an estimate of the likelihood of success against the ideal profile. But also we can look at the uncertainties in all of that underlying data, propagate it through the calculation to give us a confidence in that overall score. That helps us to determine when we can confidently distinguish between compounds. And so one way to look at it is shown here. This is what we call a snake plot in StarDrop, but essentially what we have are the compounds in a data set ordered from the best on the left to the worst on the right by that overall score. On the y-axis is the score itself. And so immediately we can see in this example, we have some compounds that have a very nice high chance of success, about 80%. That would be wonderful. And you can also see the error bar around each individual score. That shows us the confidence in the overall score given all of that underlying uncertainty in the data. And so in fact, there's lots more information here. In particular, for example, if we look at the top 5 compounds here on the left-hand side, you see their error bars overlap almost completely. And that's telling us that the data we have do not confidently distinguish between those compounds. What that means is I need another criterion to choose between them. That might be my experience or my knowledge, or maybe I need to measure that data better and get smaller error bars so I can confidently choose between them. On the other hand, here you can see that roughly the bottom 50% of compounds, the error bars don't overlap with the best. We can reject those with confidence, save our time and effort, and really focus up here where the chance of success is highest. So that's just one way we can look at these scores. This example of a profile, this is a nice one where we've got some nice high scoring compounds. But let me hand over to Samar again, who's going to tell you about what happens when we take the alchemite model that she described and the results from that and apply this probabilistic scoring approach to this large virtual library.
Thanks, Matt. So we've applied the multi-parameter scoring approach on the TP project, where we calculated the probability scores for the compounds in the virtual set based on as a number of selection criteria, covering activity against the TB assay, the solubility, hepatocyte clearance, and plasma protein binding. And what we see here from the plot for the probability score for these compounds is that no compound had a probability score higher than 0.1. In other words, there were no high-scoring compounds, which indicates that the chances of success for these compounds to meeting the criteria, the selection criteria, is very low. So we wanted to investigate why was this the case. So we first looked at the top scoring compounds, the compounds that had the highest probability scores that are presented here, and these compounds are also represented by their predicted values for the endpoints of interest. We can observe that these cells are color coded, so ranging between green to red, where the green color for a particular value means that there are high chances for that compound to meet the selection criteria for that particular endpoint. Whereas a red color means that the chances for that particular compound to meet the selection criteria for that endpoint is quite low. So because the most important selection criteria in this project was activities against the TB activity assay, we see that most of the top-ranking compounds are green in green shaded colors in the cells for the TB activities assays, the first and second column. However, these compounds seems to not meet the selection criteria for the active properties, especially for the solubility and the hepatocyte clearance. So what we would like to investigate is how the values, the predicted values in the TB activity assays, especially with regards to assay 2, would compare with the predicted values for the hepatocyte intrinsic clearance. So what the histograms we have towards the left represent the distribution of the predicted values for the TB activity assays in the top and the mouse hepatocyte intrinsic clearance in the bottom. And we see that a proportion of the bars in this histograms are colored green for both the endpoints, which means that a subset of the predicted values do meet the selection criteria if we looked at the endpoints individually. However, when we plot the predicted value for the TB activity against the hepatocyte clearance, we see that the region that corresponds to meeting both criteria simultaneously have very few compounds. We can also subset these compounds and focus on the top confident 50%, and we see that the points that predicted with least confidence is actually further away from the high activity or low clearance, which means that the model was able to predict highly active compounds and low and compounds with low clearance with high confidence. The other challenge that we've seen for the top-ranking compounds that they had solubility that were predicted to have very low chances of meeting the solubility criteria. So we wanted to investigate further how the predicted values for the TB activity assay compared with the solubility endpoint. So as we've seen before, for the histogram, which represents the distribution of predicted values for the TV activity, we've seen that there is a region where there's high chances of meeting the selection criteria. And if we looked also at the histogram for the predicted values for the solubility, we see that that there is a substantial proportion of these predicted values that have high chances of meeting or of likelihood of meeting the solubility criteria. However, if we plotted the predicted TV activity against predicted solubility, the region which represents meeting both criteria simultaneously shows that there were actually very few compounds that actually meet this criteria. And when we focus on the most confident 50% of predictions, we see that this number becomes smaller. So less compounds are actually meeting these criteria simultaneously. And these examples show us or explain to us why we were seeing very low probability scores for compounds in this virtual set. So in conclusion, the compounds in the virtual set were predicted to achieve good activity or hepatocyte stability or good passive solubility. However, it's unlikely that these compounds would be able to achieve the three criteria simultaneously. So an application of such high quality multi-parameter model enable us to explore the chemical space around the series of interest rigorously, and we can use this model to save time and resources by focusing on a small subset of compounds that for synthesis in order to validate the proposed hypothesis. So overall, we can say that unfortunately we are unable to find compounds that have high chances of meeting the selection criteria. However, it's also valuable to understand that there are low chances, and therefore we can save time and resources into running multiple experiments. And I'll hand back to you, Matt, so you can show your final thoughts on the project and the results that we've received.
Thank you very much, Samar. So just to sort of wrap up here, what Samar demonstrated is that Alchemite was used to build both imputation and virtual models on a reasonably large data set, 300,000 compounds by about 500 experimental endpoints. I think what was really nice is that we saw there was no loss of accuracy over project-specific models, even though many of the endpoints, activities, and chemistries were completely unrelated to one another. This is consistent with some other findings. We published a paper last year in collaboration with Constellation Pharmaceuticals on a smaller scale data set to demonstrate this. It's nice to see this backed up on a much larger data set and much larger range of different experimental assays, as we've shown today. And as Samar pointed out, the global virtual model was more accurate due to the additional chemical diversity that it could access in the training set. And what this means is we have a sort of a genuine build once, run everywhere model. We know we spend a lot of time in drug discovery, often building local data sets, particularly around activities, really tailored to specific series or project data sets, specific activities. And often you find when you take these deep learning methods and try to put together all the data, the presence of uncorrelated endpoints completely sort of messes it up and all of your predictions get worse. So this means we can save a lot of time. We don't need to build lots of different individual project models, and simultaneously means we can maximize the information, particularly with properties like ADME properties. We know that these global models can add a lot of information and learn across multiple projects, chemistries, and therapeutic areas simultaneously. So it's almost like the best of both worlds. Another thing that Samar focused on was this very strong agreement between the confidence that the model has in the prediction, that uncertainty estimate or error bar, and the observed accuracy. This is a really big challenge in machine learning. There was a great paper by Connor Coley's group from MIT where they tried dozens of different methods, different modeling methods, different domain of applicability methods, and were unable to find one that was repeatedly and robustly applicable. But here we've demonstrated it. Again, if you look at our other publications and webinars, you'll see this time and time again. And what this means is that even for some models that have a poor headline accuracy, we might have to throw it away using most modeling methods because we don't know what predictions are good and what predictions are bad. Even in that worst case scenario, this enables us to actually focus on valuable results that we can use for our decision making where that confidence is highest. And then Samar presented an example application to a TB project. We showed how we can combine this with multi-parameter optimization. Now, unfortunately, the result was rather unwelcome for the project. We wish we could have delivered and said, here's a great compound. But the fact that we were able to explore this very large virtual library, looking at many different optimization strategies and say, look, it's very, very unlikely you're going to achieve this balance that you require, will help to save expending time and effort with a low probability of success. Yes, you want to confirm that predicted hypothesis, but you can do that with a very small number of compounds and not continue to iterate and iterate and iterate with a very low chance of delivering a successful compound. So I must acknowledge, obviously, Samar did almost all of the actual work on the computational side here, but all of the practical work, the experimental measurements, the chemistry, the synthesis, and so on, was performed at the University of Dundee, led by Paul Wyatt at the Drug Discovery Unit, with Fabio, Laura, Simon, James, and many other colleagues in that project. Also need to acknowledge our colleagues at Intelligens, particularly Gareth and Tom, who continue to develop and improve the Alchemite method, and also our colleague Ben Irwin here at Optibrium, who also works on the applications of Alchemite and provides a lot of great guidance in these projects and helpful suggestions as well. Now, we're almost at the Q&A session. We'll be there in a couple of minutes, but I just wanted to mention that the method that you've seen today, the Alchemite method, is actually implemented in our Cerella platform. So Cerella actually deploys this method for deep learning imputation in an active learning scenario so that it can connect directly with your data sources and populate this sparse matrix of experimental data, automatically update that as new data become available so that it's always learning from the latest data. It applies Alchemite to that matrix, both in terms of imputation and virtual models, as Samar has demonstrated, and then can fill in those blanks automatically. The imputation, the fantastic results that Samar showed you, they're automatically filled in, and then it manages what we describe as the massive matrix of imputed and experimental results to make those very easy to access. Now, on the scale of a global pharma company, this can contain tens of billions of data points, and so it's developed to be very scalable, so you can search that quickly, retrieve the most interesting compounds and experimental results. And those results and the models are accessible in any platform that you wish to use via a RESTful API, of course, also in our Stardrop software. And if you want to see some examples of that in action, we had a webinar just a few weeks ago where we gave some live demos of that. So Cerella can help in many different ways. We saw this example of filling in this high quality predictions using the imputation approach. We can use this confidence estimate not only to find the most accurate predictions, but also to find hidden opportunities, experimental outliers, and potentially false negatives. We can use these models as well to get insights into the most valuable experimental data to generate in order to make better predictions downstream as well. We have an example coming up in a webinar in June in collaboration with Genentech where we can demonstrate that. And ultimately gain more value from your compound data in particularly accurately predicting complex, cell-based phenotypic or even in vivo experimental endpoints, which are very difficult to predict, if not impossible, with conventional QSAR modeling. And in this particular case, we looked at application to a global compound data set as well as project data sets. But what's really very nice is the method actually applies even if you only have a single project's data. We demonstrated that in a nice collaboration with Constellation Pharmaceuticals. So unlike many deep learning methods where you need a vast data set to get value, this imputation approach can really leverage value even in small data sets early on in a project. If you'd like to try it yourself, we can work with you in a sort of a proof of concept study. This is one example that we presented today. You'll see many other in our library of webinars. We work very collaboratively based on your projects, your data, your objectives. We also have a demo server for Cerella now with public domain data. So if you just want to kick the tires and see what it's like to work with, we can provide access to that absolutely free of charge. You can use this via our Stardrop software. If you don't already use Stardrop, we'll provide a free license during that trial period, and you can explore. It's actually a public domain data set called the Pathogen Box, published by Medicines for Malaria Ventures. Of course, the real proof of the pudding, if you like, is using your own data with Cerella, and we can deploy a specific instance so that it's completely secure. You can work with your own data. It will automatically build and update those models. You can explore them to your heart's content. And if you're interested in any of those options, please feel free to contact us at info@optibrium.com, and we'll be happy to tell you more, give you more information, and if you'd like, set up an evaluation in any one of these different ways. All right, that's enough of the sales pitch. Let's get on to the Q&A session. I can see we already have lots of questions that have come in. It's great. Just a reminder, you can ask questions via the question panel in the GoToWebinar controls. So if you haven't already asked your question and have something you really want to know, just type it in there. It'll appear here on our control panel, and we'll work through as many of those as we can in the next, 5 or 10 minutes. We'll overrun slightly if we need to. And I guess some combination of Samar and I will get through as much of this as possible. If we can't get through all the questions, we will follow up by email as soon as possible after the webinar completes. Okay, so Samar, I'm going to give you the first question here. The question here is, what is the nature of the assay endpoints in the data set that was used for training and prediction? Are they uniform? Are they only sort of EC50 or MICs, or are they different? Protein level, organism level, cell level, MICs, EC50s, etc.
Yeah, thanks, Matt. So we in this data set there were heterogeneous type of data. So we've seen percentages, IC50, MIC. So yeah, it was a wide range of endpoints with different units.
Yeah, absolutely. And also, phenotypic and cell-based, organism level, things like anti-malarial, as well as target-based activities, a real mix of different types of data, and you can build a single model across all of those. I can see we've got a couple of questions here that are closely related. So let me see if I can try and address both of them simultaneously. One person says, okay, global is better, but even though it's sparse, the global data set has more data points than the project data point. And so therefore more data points should result in better models, at least usually. Another one that's related is that the value of global models, especially for ADME endpoint, is well reported to offer significant advantages over local models. What were the findings of the current analysis with respect to potency prediction? How did the global model or the model including multiple endpoints compare with the local model for potency endpoints? So those two go hand in hand. One would like to think that more data is always a better model. But what you do often find, particularly for activity endpoints, is that actually we need to build local models using conventional QSAR approaches to get the resolution that we need within a single series or a single project. And I think what was particularly powerful here is that, yes, of course, we expect to see some improvements as we did in the virtual model for sort of ADME endpoints, but even for the the project-specific endpoints, the target activities, and so on, we didn't need to build a local model. We didn't lose any information by trying to build models over diverse chemistry and completely unrelated endpoints. So where you expect to gain an advantage, that advantage was gained. Where you expected to lose accuracy, as you would with a conventional QSAR approach, the accuracy was actually retained. This gives a sort of build once, use everywhere sort of outcome that we mentioned. I hope that addresses the question. Here we go. Let's see. What was the strategy used to generate the test set? Was it a random split, scaffold based? How was that done, Samar?
Yep. So in this case, we've run a random compound split, but we've tried to make sure that the proportions of measured values for each endpoints in the training and the test are the same. So although, yeah, we've run a random compound split, but we've tried, yeah, to maintain the proportions.
Fantastic. Thank you very much. Another great question here. The descriptors are physical properties and other fingerprints of the compound, but some ADME parameters depend on recognition of specific molecular features. For example, PGP recognizes secondary amides. How do you include the molecular recognition along with the physical properties? So that's in terms of the descriptors that we use. So the descriptors we use are actually the ones that we use within our Stardrop auto modeler. So they absolutely include physicochemical properties and sort of whole molecule properties like log P, McGowan's volume, charge, flexibility, aromaticity, and so on. But also particular functional groups and their relationships to one another encoded as smarts. So we do include in our library of descriptors, particular functional groups that are known to sort of be key recognition features for many endpoints. I should say that in this particular example, we used only 2D descriptors, and in fact, one interesting thing is to combine this with maybe more expensive sort of 3D calculations or simulations. One of the nice things is that you can take sparse data as input. So even if you haven't run your simulation on every single compound, you can still use that and recognize the relationships and the correlations between those simulations and experimental values to improve the accuracy of your predictions. We didn't do that in this case, but it's another great example of how we can extend it and use other forms of descriptors. Okay, here's another one for you, Samar. I'm not sure how much you can say about this, but what is the similarity distribution in the global set?
Yeah, so they were like various, like multiple chemical spaces, so like multiple clusters of chemical spaces, because they were collected from multiple projects. So we would see like within a single project, the compounds are more similar, and then across projects, the similarity decreases.
Yeah, absolutely. I mean, this was from a very large number of projects across lots of chemistry. Unfortunately, we don't get to see the structures. This is because it was done in collaboration. The structures were all anonymized by our collaborators at the University of Dundee, but we do know they covered a very broad range of chemistry across different therapeutic indications and so forth. So,
yeah. So we are only able to look at the correlations in the descriptor space. But not necessarily looking at the structures themselves. So, yeah.
Yeah. Another great question here. So I'll take this one. Is the estimate of the confidence and uncertainty based on just the closeness of the compound to the training set? It's a great question. Yes and no. Obviously, proximity to the training set is one important factor that's taken into account. Have we seen similar compounds before? But the beauty of Alchemite is taking not only chemical descriptors, but that sparse assay data. So it's not only trying to interpolate we're extrapolating in structural space, but also in sort of bioactivity space simultaneously. So it's also to do with what experimental data are available for a particular compound and how relevant that data is to predicting the missing values. And it's also related to the variability in that experimental data itself. So multiple factors come together, and that's how we're able to get this much better estimate of the uncertainty and hence accuracy of those individual values. It has another very nice effect, which is because it's using that biological activity space and property space at the same time, it does actually extrapolate better in chemical space than conventional QSAR methods. We demonstrated that in our paper in actually 2 papers, one in 2019, excuse me, the other in 2020 in Journal of Chemical Information and Modeling, where we looked at that extrapolative behavior. We have another one that we'll be submitting soon, looking at activity cliffs and its ability to better detect those as well. But you'll have to stay tuned for that webinar and publication. Right, I'm aware that we really have gone quite a bit over time. There's one very good question here about the project specifically. So let me just address that. It says, for the TB multi-parameter optimization, what is the reason to include protein binding as a key parameter? How was the desired range for it determined? Also, both hepatocyte clearance and microsome clearance, you'd expect those to be related. So surely hepatocyte clearance would capture clearance by microsomes. What's the consideration for including both for the MPO? Both are excellent questions. I guess the starting point is the MPO profile was not our choice. This is what we were asked to use by the project. The key reason for using both hepatocyte and microsome clearance is that it actually was a very low correlation between those. So they wanted to include both of those in the profile. They were finding compounds that were highly stable in microsomes and very unstable in hepatocytes. I guess one might argue that you'd expect the unstable compounds in microsomes to also be unstable in hepatocytes, but they wanted to look at the 2 different factors independently. And the protein binding, I guess there's lots of discussion about the relevance of protein binding as a parameter, but they felt they wanted a low protein binding in their animal model to get good exposure for the unbound drug. Again, opinions may vary there, but that was their rationale. Okay, well, I thank you to everyone who stayed on so far past the hour. I can still see there's lots of you, but I think we'll probably draw the proceedings to a close. And finally, just again to say a big thank you for joining us today. I hope that you've enjoyed the webinar. If you want more information, any questions we haven't addressed, we'll get back to you, but if something occurs after the fact, please do email us at info@optibrium.com. And just a reminder that there will be a recording posted very shortly on the Optibrium website at optibrium.com/community. So if you want to refresh your memory or indeed share the webinar with any of your colleagues, please do feel free to pass on the link. So thank you, Smar, for a great presentation. Thank you all for joining us, and we look forward to seeing you again at a webinar soon. And who knows, someday, maybe even in person. So thanks a lot.